Hello, thank you all for coming. My name is Nate Geip, and I'm going to be presenting the first section of a collaborative research project between myself, Lily Erickson, and Charlie Adams. Our study is collectively titled The Influence of Local Habitat on Insect Communities. Lily and Charlie are going to tell you about the insects, but before you hear about that, you need to hear about the local habitats part of that title. That's the focus of my study. Specifically, I study the plant and soil characteristics of these local habitats, so my section of the study is titled A Comparison of Plant Assemblages and Soil Analyses in Two Forest Habitats. The relationship between plant and insect biodiversity is what unites this entire study. It is well established that plant species richness is positively correlated with insect species richness. The more species of plants in a habitat, the more species of insects you will find there. This makes intuitive sense. A rainforest has more insect species than a forest in, say, Hillsdale. But is plant biodiversity directly affecting insect biodiversity, or are they both responding to extrinsic abiotic factors? Factors like climate, location, and elevation could be confounding variables in this relationship. Going back to our example, does the rainforest have more insect species because there's more plant species than in Hillsdale? Or does the rainforest have more plant and insect species because it's sunnier than Hillsdale? It is hard to set up an experiment to answer this question because you would need to have two habitats with radically different plant assemblages but the same abiotic factors. This situation is not normally found in nature. This is where the two different habitats we studied at Hillsdale's G.H. Gordon Biological Station come in. This photo is an aerial view of the biostation. You can see Rockwell Lake is labeled there, and this is where the two habitats we studied are located. I'll be referring to this one as the dredge forest, and this one as the hardwood forest. Throughout the presentation, the dredge forest will be represented with blue and the hardwood forest with yellow. You can see that these two habitats are geographically adjacent. This is important because it means that extrinsic abiotic factors like climate, location, and elevation are naturally controlled for. They are basically the same for both forests. This gives us the opportunity to study the plant and insect communities in these habitats while minimizing the influence of these abiotic factors. While the extrinsic factors in these habitats are essentially identical, they have very different histories which have cultivated what seem to be very different plant communities. Prior to the 1900s, much of northern Michigan was covered by white pine forests. When these forests were logged, many were replaced by deciduous forests of oak and maple. The hardwood forest at the biostation is one such forest. It is a mature forest that has been around for much longer than the dredge forest. On the other hand, the dredge forest is much younger and is at an earlier stage of succession. Unlike the hardwood forest, the dredge forest is still undergoing large changes. To understand why this is, we need to look at the dredge forest's history. In 2001, Rockwell Lake was dredged in order to make the lake deeper. The sediment that had accumulated on the bottom of the lake, called dredge, was removed and pumped into a depression formed by a railroad grade. That's this trapezoidal shape here, the future site of the dredge forest. Because the area was flooded, all plant life was destroyed. This set the stage for primary succession, the process by which plants begin to colonize an area. By 2003, the dredge had dried significantly, and natural colonization created the marshland environment shown here. This picture was taken by a previous Hillsdale biology student, Tanya Eaton Frazier, during a study that she did on this habitat. I'll be talking about her study later in my presentation. By the summer of 2019, when I did my research, the dredge was at a later stage of succession. Shrubs and small trees have succeeded the marshland plants. It's now no longer a marsh, but a forest. So in summary, we have two habitats with what seem to be very different plant communities in close proximity to each other. This means things like weather and location are controlled for. This setup is unique because it gives us a chance to study the relationship between the plant and insect communities in these habitats with minimal interference from extrinsic abiotic factors. The purpose of my section of the study is to characterize and compare the understory vascular plant assemblages and soil properties in these two habitats. Ultimately, this will provide base data to study the relationship between plant and insect assemblages. To accomplish my purpose, I selected 42 1 by 1 meter squares in each habitat, called quadrats. To do this, I first superimposed 5 100 by 100 meter squares on each habitat using Google Earth. Each of these large squares was broken into a grid of 100 10 by 10 meter squares, which I called plots. I numbered the plots 1 through 500 and then used a random number generator to select 6 in each habitat. Each plot was then broken into a grid of 100 1 by 1 meter squares. These were the quadrats. The quadrats were numbered and 7 were selected from each plot. 7 quadrats in each of the 6 plots meant that there were 42 quadrats in each habitat, for a total of 84 quadrats in the entire study. This is what the plot and quadrat setup looked like in the field. Using Google Earth, I recorded the GPS coordinates of each of the corners of my plots. I found these points in the field using a GPS and then marked them with T-posts and strung string around the T-posts to mark the edges of the plot. This is shown in yellow. Using the edges of the plot and a tape measure, I located the quadrats within each plot and marked their corners with orange yard flags. I highlighted 2 of the more visible quadrats in this plot in red. When I surveyed a quadrat, I placed a 1 meter by 1 meter PVC pipe frame around the corner flags. I then identified all the vascular plants present in the quadrat down to the genus level. Using cardboard cutouts of standard areas and a ruler, I estimated the area each genus of plant covered in square centimeters. I also estimated the total area of the quadrat covered by plants. In addition, I used a trowel to collect a specimen of every genus I found, which I put in the plant press for preservation. These specimens will be mounted and saved in the Hillsdale College herbarium for future research. I randomly selected 6 quadrats within each habitat to sample with the soil probe. The soil probe is a hollow tube you shove into the ground to obtain a soil profile. I sampled one 15-centimeter deep core from all 12 of the selected quadrats, placed the samples in Ziploc bags, and froze them so that they could be transported back to Hillsdale for lab analysis. I also selected an additional 5 quadrats from each habitat to sample for nutrient analysis. I took a pint of dirt from the top 15 centimeters of these quadrats using the soil probe. These samples were frozen and then taken to the Soil and Plant Nutrient Lab at Michigan State University for nutrient analysis. Back at the botany lab at Hillsdale, I thawed my samples for 1 hour and squeezed all the air out of the bags. I submerged the bags in a graduated cylinder of water to measure volume, and then transferred them to a Pyrex dish to measure their initial mass. These measurements would be used to calculate the sample density. Next, I put my samples in an oven at 110 degrees Celsius for 16 hours to evaporate any water present. I weighed them afterwards to determine the percent water content. I then put my samples in a muffle furnace at 400 degrees Celsius for another 16 hours to burn off any organic material. I weighed them afterwards to determine the percent organic content. After the water and organic matter had been removed from the samples, what remained was the sediment. Sediment is the bits and pieces of eroded rock in the soil, what you think of when you think of dirt. Sediment can be separated by particle size. To do this, I put my sediment through a sieve tower. The sieve tower is a stack of dishes, each with wire mesh at the bottom. This picture shows what a single sieve looks like. As you go down the sieve tower, the mesh in each sieve gets finer and finer. Sediment retained on the first sieve is called gravel. These are the biggest particles. Next is sand, then silt, and anything that goes to the last sieve is clay. Clay particles are the smallest. I shook each sample through the sieve tower for 5 minutes to separate it into its gravel, sand, silt, and clay fractions. Each fraction was then weighed to determine the composition of the sample's sediment. Before I discuss my plant results, it is worth noting that some specimens I encountered were either too small or lacked enough identifying features to identify down to the genus level. These plants were put in the category Other and excluded from the study. Of the 84 quadrats, 20 contained specimens in the category Other, but in these quadrats, the average coverage was less than 0.25%, so these plants are essentially negligible. Genus richness is the number of plant genera found in each habitat. Of the 44 total plant genera found, 26 were unique to the dredge forest, 9 were unique to the hardwood forest, and 9 were found in both forests. As this pie chart shows, the dredge forest has a higher genus richness than the hardwood forest. Genus biodiversity takes into account genus richness and evenness. Evenness is the degree to which specimens are evenly distributed among the genera present. A higher evenness indicates a more equal distribution and a higher biodiversity. The Shannon-Weaver and Simpson diversity indices are the 2 most common ways to measure biodiversity. I used the percent coverage data and the Shannon-Weaver and Simpson diversity equations to calculate the genus biodiversity for each habitat. No matter which equation you use, the dredge forest has a significantly greater plant genus biodiversity than the hardwood forest. 13 genera were found in more than 10% of the quadrats in the dredge forest. The three most common genera were Solidago, or goldenrod species, Salix, or willow species, and Populus, a genus which encompasses aspen and poplar trees. A quaking aspen is pictured here. Three genera in particular will be important later in Lily and Charlie's study. They are Salix again, Geum, also known as Avens, and Lonicera, or honeysuckle. 7 genera were found in more than 10% of the quadrats in the hardwood forest. The three most common were Pteridium, a genus of ferns, Acer, or maple species, and Carex, a genus of sedges. The genus that will be important in Lily and Charlie's study in this habitat is Pteridium, the ferns. Of all 44 genera in both habitats, the three most common were Acer, Pteridium, and Solidago. You'll notice that there are only 9 bars that are both blue and yellow, meaning that this genus was found in both the hardwood and dredge forests. Of these genera, most are skewed heavily toward one habitat or the other. There is little overlap between the habitats. This indicates that the plant assemblages in either habitat are composed of very different genera. This is a non-metric multidimensional scaling ordination, or NMS. Each point on the graph represents a quadrat in my study. I removed their labels to make the graph easier to read. The closer 2 points are, the more similar the plant genera in those 2 quadrats. The axes in this graph are constructed and don't represent specific variables. What's important is the distance between points. Like the previous graph, the NMS demonstrates that the hardwood and dredge forests are composed of distinct plant genera. You'll notice the dredge forest quadrats are grouped together and the hardwood forest quadrats are grouped together in distinct clouds. These two groupings are very far apart, meaning the two habitats have very distinct plant genera. In addition, the dredge forest cloud is more spread out than the hardwood forest cloud. This suggests that the dredge forest has greater differences within its quadrats than the hardwood forests. This is consistent with the fact that the dredge forest has a higher biodiversity. To determine the effectiveness of my plant sampling method, I constructed a genera versus area sampled graph. The y-axis is the cumulative number of genera found. The x-axis is the cumulative area sampled, or number of quadrats sampled. The blue line is the actual number of genera I found in the dredge forest. This line is constructed using a technique called bootstrapping. A program graphs the cumulative genera found in the 42 dredge forest quadrats for every possible order those quadrats could be sampled in, and then takes the average of all of these lines. If I did not use bootstrapping, this line would be arbitrary, since it would depend completely on the order in which I sampled my quadrats in. The green and red lines are coverage-based estimators. There are 2 different ways of estimating the actual number of genera in the dredge forest based on the number of rare genera I found. According to the average of these estimators, I found 54% of the genera present in the dredge forest. Considering I sampled less than 0.08% of the forest, that's not too bad, but more sampling is definitely necessary to find the remaining plant genera. This is the same graph, but generated for the hardwood forest. The yellow line is the number of genera I actually found, constructed with the bootstrapping technique, and the green and red lines are the 2 estimators again. According to the average of these estimators, I found 81% of the genera in the hardwood forest. More sampling would be useful in this habitat as well. Since I sampled the same number of quadrats in each habitat, my sampling effort in each habitat was the same. However, the same effort in the dredge forest found a much lower percentage of the total genera present than it did in the hardwood forest. This also indicates that there are more genera present in the dredge forest, and that it has a higher biodiversity than the hardwood forest. Before I move on to my soil analyses, I want to briefly compare some of my plant results with the results of Tanya, who I mentioned at the beginning of the presentation. This is Tanya in the dredge forest in 2003. She called it Gordon's March, which was a much more apt name at that point. You can see she's wearing snowshoes because in 2003 you could actually break through the top layer of dirt in the dredge and fall into the layers of semi-solid muck below. She's also tethered to the edge of the habitat, That way, if she does fall through, someone can pull her out. So it was a very different habitat back then. Tanya was interested in comparing Gorin's Marsh to two other wetland habitats on the biostation's property. She sampled plants in all three of these habitats using a very similar method as me. You can see she's got her 1 by 1 meter PVC quadrat. She found that Gorin's Marsh had the fewest shared species of all three of the habitats. Even back then, it was a very unique habitat. The most common species she found there was cattail, which she is surrounded by in this picture. I did not find any cattail, which is evidence that the dredge forest plant assemblage has been changing. Of the specimens she collected from Gorin's Marsh that are in the Hillsdale College Herbarium, 3 genera are still present in the dredge forest today: Salix and Carex, which you've already seen pictures of, and the genus Hypericum, which is also known as St. John's wort. 3 additional genera that Tanya found in her study but did not collect specimens of in Gorin's Marsh were Asclepias, or milkweed, Fragaria, or strawberry, and Rubus, or raspberry. I found all 3 of these genera in the dredge forest in 2019, which is further evidence that succession is occurring and the dredge forest plant assemblage is changing. Moving on to the results of my soil analysis. Simply based on the qualitative analysis of these 2 pictures, we can see that the soils in these 2 habitats are very different in both color and consistency. The darker color of the dredge soil suggests that it is higher in organic content, while the brown color of the hardwood soil suggests that it is very sandy. I separated my soil samples into the components of water, organic content, and sediment. Since each component is measured as a percentage of the total weight of the sample, I used an arcsign transformation on the percentages so that I could run a t-test. The dredge soil contains a significantly greater percentage of water and organic content than the hardwood soil. This makes sense given the dredge forest's history. It used to be a marsh, and the soil comes from the bottom of a lake, where organic material from everything that dies in the lake accumulates. The hardwood soil has significantly more sediment than the dredge soil. These pie charts show the composition of the sediment fraction of my soil samples. This data was obtained by sieving the samples, so you'll see the different particle fractions I mentioned earlier listed in the key. A small amount of gravel was present only in the hardwood soil, and neither forest contained any clay. A small percentage of all samples was lost in transferring the fractions from the sieves to the Pyrex dishes to be weighed. The percentages of silt and sand in both habitat sediment are relatively similar. Essentially, the dredge soil looks like the hardwood soil, except for the fact that someone added a large quantity of water and organic material. This is essentially what happened when the dredged material from the lake was dumped into the habitat. Using the initial weight and volume of my samples, I was able to compare soil density between the habitats. The hardwood soil is significantly more dense than the dredge soil. The dredge soil density is less than 1, which means it is less dense than water and should float. This makes sense if we go back to the dredge forest history. As the dredge in the dredge forest dried, a layer of soil solidified on top of the layers of semi-solid muck underneath. This dried layer sort of floated on top of the muck underneath it. You can still see this today, where there are sections of the dredge forest that are almost bouncy, like a trampoline, because of this phenomenon. So it makes sense that the soil from the top layer of the dredge forest has a density less than water. The pH of the dredge soil is significantly greater than the pH of the hardwood soil. The hardwood soil is likely more acidic due to rainwater washing nutrients out of the soil, which is known as leaching. Sandy soils are more prone to leaching since they drain rainwater very well. The hardwood soil is sandy, so rainwater drains through it quickly and carries away nutrients like magnesium and calcium, which can help prevent soil from acidifying. The data displayed in these next graphs came from the MSU Soil and Plant Nutrient Lab. I originally planned to test 10 samples, 5 in each habitat, but 1 sample from the hardwood forest was contaminated in transport and had to be discarded. These graphs show the levels of various plant nutrients in the soil, measured in parts per million. The dredge soil contains significantly more magnesium, nitrate, and ammonia than the hardwood soil. This is consistent with the leaching that I hypothesize is going on in the hardwood forest. Michigan State also measured the levels of calcium, potassium, and phosphorus in the soils. Like the previous nutrients, the dredge soil contains significantly more calcium than the hardwood soil. The high levels of calcium could be contributing to the basicity of the dredge soil. they could indicate the presence of high levels of calcium carbonate, which is a basic substance and also a buffer. Calcium carbonate would make the dredge soil more basic and cause it to resist changes in pH. Interestingly, there is not a significant difference in the levels of potassium in either soil. Earthworms are known to increase potassium levels in soils, so the presence of earthworms in the hardwood soil could be a possible explanation for this result. Also interestingly, the hardwood forest has significantly greater levels of phosphorus than the dredge forest. A possible explanation for this is the acidity of the hardwood soil. In acidic soil, phosphorus becomes protonated, and plants cannot absorb this protonated form. So possibly the low pH of the hardwood soil keeps the plants from absorbing large amounts of phosphorus, so it remains in the soil. In conclusion, the plant community in the dredge forest has significantly higher genera richness and biodiversity than the hardwood forest. This is likely due to some combination of 3 different factors, all of which happen to begin with S: soil, succession, and sunlight. It's possible that because the soil of the dredge forest is, with few exceptions, more nutrient-rich than the hardwood forest, it is able to support a higher plant biodiversity. Since the dredge soil is less sandy, nutrients are not leached away by rainwater as fast as they are in the hardwood forest. In addition, the high organic content in the dredge from the time it spent at the bottom of the lake could also be an explanation for the high levels of nutrients. The next S is succession. The hardwood forest is farther along in its successional history than the dredge forest. Habitats in earlier stages of succession generally have a higher plant biodiversity because new plants are always moving in to colonize the area. Lastly, sunlight. The hardwood forest is almost completely shaded by tall maple and oak trees. Most of the tall plants in the dredge forest are shrubs and young trees, so there is more sunlight reaching the ground. The greater availability of sunlight in the dredge forest may contribute to its ability to support a higher plant diversity than the hardwood forest.